Welcome to a tour of the Georgia State University and Georgia Institute of Technology Joint Center for Advanced Brain Imaging. This event is sponsored by the Tri-Institutional Center for Translational Research in Neuroimaging and Data Science, or the TREN Center. We are a collaborative research center that combines expertise across Georgia State University, the Georgia Institute of Technology, and Emory University. Find out more about our research at trendcenter.org. This event was also sponsored by Brain Awareness Week via the Dana Foundation.
Hello, my name is Dr. Nikki Arrington. I am the Supervisor of Research Operations here at the Georgia State University, Georgia Institute of Technology Joint Center for Advanced Brain Imaging, better known as CABI. At CABI, we seek to advance scientific discovery and theoretical innovation in brain science by providing cutting-edge neuroimaging, non-invasive brain stimulation, and analytics tools through multi-institutional collaboration. Our research facility allows scientists to collect neuroimaging data to answer their research questions. But you might be wondering, what is a neuroimage? And how do you collect this type of data? Hopefully, by the end of this video, you will have the answer to these questions and more. The word neuro means relating to the nerves or the nervous system and the brain. So a neuroimage is a picture of the brain. But how do you take a picture of your brain when it is hidden inside your skull? Doctors and researchers both can use a very special camera called a magnetic resonance imaging or MRI scanner to take these types of images. An MRI uses powerful electromagnetic and radio frequency waves that are able to safely and painlessly penetrate below your scalp and skull to reveal different aspects of your brain's structure and function. At CABI, many researchers utilize our state-of-the-art MRI scanner to answer questions about the brain. While many people may have received an MRI scan at a hospital, CABI is a little bit different in that researchers here do not diagnose or treat disease. Instead, they use different types of neuroimages to better understand how the brain develops or is involved in different activities. These different types of images are called imaging modalities. Let's take a look at the different type of imaging modalities and how they're used by researchers here at CABI.
Hi, my name is Michaela Jetter, and I am a graduate student at Georgia State University, and I use structural MRI. Structural MRI provides information on the shape, size, and integrity of brain structures. Structural MRI is often used in medicine to detect brain abnormalities such as tumors or neural atrophy or cell death. In research, we can use structural MRI to look at how brain structure changes across the lifespan, compare brain structures across individuals with and without brain diseases, and determine how brain regions might grow or change if we engage in healthy brain activities, such as problem solving or exercise. In my research, I use structural MRI to answer questions about the size of different brain structures and how they impact our reading and attention abilities.
My name is Dr. Turner, and I use functional MRI. Functional MRI is different from structural MRI in that we use functional MRI to look at brain activity rather than the structure of the brain, even though we use the same MRI machine for both possibilities. So functional MRI relies on the magnetic properties of the iron in the blood to measure oxygen use across different regions of the brain. It works on the fact that as neurons fire, as the brain is functioning, as neurons fire, more oxygen flows into that area, which changes the properties of the iron in the blood, and that actually makes a paramagnetic measurement or paramagnetic change that we can then measure using functional MRI. So the more a region uses oxygen, the more active it is, the more we can actually see changes over time. And by over time, we mean over the course of a few seconds. Because its use is fairly new, Functional MRI is still primarily used in research, though clinically it has been used quite a lot in pre-surgical mapping. So for example, you might, if you're going to do surgery on someone to remove a brain tumor or to try to deal with an epilepsy problem, you might do all sorts of functional MRI scans first to determine what parts of the brain are likely to be affected and what cognitive processes they tend to be active during, so that you know what kinds of deficits the person may need help with as they move forward. But it is slowly being incorporated more and more into diagnosis and tracking of neural diseases and disorders that impact brain activity. And that ranges from childhood developmental issues to Alzheimer's disease and dementia, depression, neuropsychiatric disorders, you name it. In research, we can use functional MRI to look at how brain activity changes across the lifespan, how brain regions act together to support cognitive or thinking processes, emotional processes, how it changes in response to experience or treatments, etc. In my research, I use functional MRI to answer questions about why some people hear voices as they're developing schizophrenia, or why depression may look different in different groups of people across the brain, and other related questions. I hope you've enjoyed what you're finding out about the Neuroimaging Center.
Hi, I'm Eric Semmel. I'm a PhD candidate at Georgia State University, where I study clinical neuropsychology and cognitive neuroscience in Dr. Tricia King's lab. As part of my research with MRI, I've used a method called diffusion tensor imaging, which is also known as DTI. DTI is interesting because it actually lets us visualize the white matter tracts in the brain. So if you think of your brain as a computer, the white matter tracts are sort of like the wires that send information between different parts of the brain and allow different parts of the brain's gray matter to communicate with each other. So to obtain these images, DTI actually looks at water molecule diffusion throughout different parts of the brain. And that just means the movement of water molecules throughout brain tissue. White matter tracts are covered by a fatty substance called myelin, which serves as a kind of insulation. And water molecules have a hard time diffusing through this myelin to the outside of the cell. So because of this, they're more likely to move sort of up and down along the white matter tracts, along the white matter pathways, instead of going out and away from the white matter pathways. So if we look at how water moves around in the brain, we can create a picture of our brain's white matter structure because of all of this. So in my research, I've used DTI to look at the white matter in survivors of pediatric brain tumors. Brain tumor patients are at risk for differences in their white matter structure due to the presence of the tumor itself, as well as some of the life-saving treatments they get, like radiation therapy. Our goal in studying this population with DTI is to look for brain-based predictors that an individual might have more trouble thinking, remembering things, taking care of themselves, and things like that later on in life, so that we can hopefully intervene earlier and provide services and therapies that they need.
In addition to my role here at CABI, I am also a researcher at Georgia State University. I use neuroimaging and transcranial magnetic stimulation to better understand how the brain is involved in learning to read. Transcranial magnetic stimulation, or TMS, is one step beyond neuroimaging methods such as MRI. Using what we have learned about the brain's structure and function from different types of imaging modalities, we can use a special electromagnet to influence the way the brain functions. TMS allows us to target a specific region of the brain and temporarily facilitate or suppress the brain's activity. TMS has been used to treat depression, obsessive compulsive disorder, PTSD, and addiction. In my research, I use TMS to impact learning and reading skills.
Hello, my name is Muhammad Sendi. I am a PhD student at Georgia Tech and Emory University. Today, I'm going to talk about EEG, or electroencephalogram. As you know, When neuron fires together, they generate a brainwave. That brainwave can travel across the brain, and when you put electrode on the scalp, you can measure that brain activity. That's called EEG. The EEG signal is different in sleep versus the wakefulness. Also, the EEG signal is different in the patient brain when we compare that with the healthy brain. Therefore, the EEG signal can be used as a brain signature when we want to study the patient brain in different neuropsychiatric or neurological disorders. The main advantage of the EEG or MRI is its huge temporal resolution. That means you can measure many brain activity in a fraction of a second. In my research, I use EEG signal. I record EEG before and after transcranial magnetic stimulation in patients with post-traumatic stress disorder or PTSD. I record pre-TMS and post-TMS signal and see how the TMS can change the EEG signal dynamics.
We hope you enjoyed this video where we discuss the types of images and brain stimulation that we collect and administer at CABI, including structural MRI, functional MRI, DTI, TMS, and EEG. We would not be able to collect all of this data without participation in our research from people like you. Would you like to be a research participant so we can look at your neuroimages and use them as valuable data in our scientific studies? If so, be sure to check out the CABI website at cabi.gsu.edu/studyvolunteers to see if you qualify. Thank you for joining us, and be sure to stay tuned for our live question and answer session.